This video is integrating signaling pathways. This is figure 16-40 from your textbook, and it's titled The Integration of Signaling Cascades. We could also substitute the word crosstalk for integration, and we can also substitute the word pathways instead of cascades. And the basic idea is that different signaling cascades can play off one another, and that the reality is that inside a cell there are many receptor proteins, and that those outputs from the signaling pathways may kind of work off each other, sometimes reinforcing the same message and sometimes presenting different messages to the cell. So the goal of this topic is to understand a little bit about what that looks like as far as this crosstalk and how a cell might determine the outcome based on sometimes conflicting information. So let's check out the figure here. Basically, you should recognize this ECR, enzyme coupled receptor, and you should also recognize the GPCR over here. And so notice that each of them in this drawing are bound by their respective signals, and then you can see the downstream events that can take place. And so you should be able to recognize these. Here we're checking out an activated G protein, which can activate adenylyl cyclase and cyclic AMP and lead to the activation of pKa. On this other pathway, we see that PLC is activated. And as you know, then we have these two different products, IP3 and DAG, which converge together on PKC down here. We haven't talked about this part here, so you can kind of ignore that. And the goal is not that you memorize this whole shenanigans and be able to recapitulate it, but to see how there's going to be several different inputs that take place. In a signaling event that we haven't talked about, the enzyme-coupled receptors can also activate phospholipase C, and then can lead to the same process to eventually activate PKC. We've just talked about the RAS monomeric G protein, which is activated by the GEF. So RAS is right here, and it can activate the MAP kinase cascade, and alternatively, RTKs can activate P13K which activates the PIP3. We didn't talk about this step, but does activate the AKT kinase. So the crosstalk portion of this figure has to do with the fact that each of these receptors, when activated, can activate the same product. Also, the ultimate output of what each of these different spots is could also potentially reinforce one another or conflict one another. And the big idea here is to see that the signaling pathways can influence one another. This figure, which is 16-43, is also from your textbook, and so it talks about the integration of signaling. There are 4 different receptors, each with their own ligand. So we see all 4 of these up here. And then each of them has a distinct ligand, which has a different shape, color, and letter associated with it. And then we can see that some of them activate the same enzymes, like A and B, both activate kinase 1. On the other hand, this ligand C, through its receptor, will repress that kinase. And then on the other hand, C and D ligands have alternate roles as far as how they influence kinase 2. C is repressing kinase 2, whereas D is activating it. And then also we can see here that ligand B, through its receptor, can also activate kinase 2. These two kinases are going to potentially influence this target, the same target protein, which you can see has phosphate groups added to it, and that would be an obvious result of the kinase activity. And so here's a little summary of what I've just talked through. That there's 4 different receptors, each has their own unique ligand. They can each affect the activity level of kinase 1 and or kinase 2, and then they are convergent, these 2 kinases, upon the same target protein. And so this is where the integration comes in, is that this target protein integrates information about activity levels from kinase 1 and kinase 2 and those activity levels are a result of the presence of ligands for each of these 4 receptors. All of this, though, all this information up here converges upon one cellular response. So one of the ways that this information can be integrated is that each of these kinases may phosphorylate one site on the target protein. And so let's take that as an assumption. And then kinase 2 phosphorylates the second site. And so to talk through this, let's say that we've got our ligand B, through its receptor, will activate both kinase 1 and kinase 2. Since each of these, in our example, are phosphorylating the 2 sites that are needed to activate the target protein, then just with ligand B, we can elicit this cellular response. On the other hand, if we check out a different combination, we can activate kinase 1 through the ligand A, but we would then need ligand D to activate kinase 2. And so if ligand B were not available, another way to elicit the same response would be to have ligand A plus ligand D. Additionally, as you can see here, ligand C represses the kinases. So we also would assume for this activity level that ligand C would not be present. And so the goal of this is to be able to interpret or talk through these series of activations and repressions, and so take a similar figure and interpret what it would take to activate a target protein. And you'd have to have this information about the phosphorylation sites and which kinase would be performing that. And so to sum this up, the idea of this integration of signaling is that the net outcome of the cell response depends on the number of signaling molecules and their regulatory role or roles. Finally, I want to show you a little more of the reality of cell signaling. And so this is nothing to memorize, but if you were provided with an image such as this, to be able to pull out what receptors are present. For example, you might recognize the insulin receptor as a RTK, since this is a growth factor, and you can see the dimer, which is present here. You can also see that there's GRB2, which you may recall is an adapter protein for RTKs. Also, kind of looking at this figure, notice that we've got a nuclear membrane here, so the nucleus, and you can see that there are a variety of different transcription factors which are queued up. Down here towards the bottom, you can see some of the outputs of this particular signaling pathway, which includes growth through this pathway and apoptosis through these pathways here. And you can see there's a dashed lines that some things may have 2 different ways to affect a signaling pathway, which is a little wild. Overall, this is to impress upon you that we've kind of just hit the tip of the iceberg for what cell signaling is. We've maybe looked at this portion of it, but know that there's a whole lot that's lurking inside the cell as we have this integration of signaling pathways and crosstalk amongst them. And the overall goal is that you should be able to pull out some pieces of information from a complex pathway such as this one.